I'd now like to share with you a short video by Dr. Stephen Manos from the Australian BioCommons, who discusses the Threatened Species Initiative Demonstrator Project and building trust and identity.
Hi everyone, thanks for having us here at the AAF Trust and Identity session, and I'm here to tell you about a project that we've been working on, which is the Threatened Species Initiative Demonstrator Project over the last year and a half or so. So we as the BioCommons engage with a number of communities nationally, and one of our key ones over the last few years has been the Threatened Species Initiative. One of the things that researchers, conservationists, biologists, and other people who, broadly speaking, are not computational or analytics experts need to conduct complex analysis. They need to be able to use tools to conduct their assembly and annotation of genomes of threatened species like the Regent honeyeater, like koalas, and so on. So as a person or as a researcher who's part of the TSI consortium, we've engaged with TSI to develop a number of user stories, a number of scenarios which they want to be able to execute. So for example, that as a researcher, I want to be able to assemble a genome assembly in Galaxy Australia based on a sequence file and a transcript file from the BPA data portal. I want to be able to automatically annotate an assembled genome in Galaxy Australia. I want to be able to ultimately share that annotated genome so I can improve that annotation and description of the genome in collaboration with my colleagues. And eventually, I want to be able to publish that draft assembly and annotation in the BPA data portal, so which ultimately means that I can share those findings with the consortium nationally and internationally. One of the starting points for our work with the Threatened Species Initiative was this diagram that came from Carolyn Hoggs, who's the lead of that initiative, that consortium nationally. This, to some of you, many of you, may look like a complicated picture, and certainly to Carolyn Hoggs it was. And unfortunately, this is the sort of picture that many researchers are left to their own devices to decipher and figure out nationally. What this picture describes is a range of different platforms that someone who needs to work effectively in the threatened species space would need to figure out themselves. They would need to understand where the data portal is, how to get to it. They would need to understand what Galaxy Australia is, how to get to it, how to get to other tools such as those that are present on this thing called the TSI Conservation Genomics Hub. What is PAWSI? How do I get there? And how do I utilize it? There's the Australian Reference Genome Atlas, which provides a view of lots of different data resources. And ultimately, we need to be able to publish these completed assembly and annotated genomes to AWS and other resources beyond, such as NCBI. From a researcher's perspective, this is a very, very complicated picture. And the question is, how can we make this picture of research, which is what the reality is today, much easier particularly for people who aren't specialists in computing to handle? So we took on the challenge between BioCommons and AAF to try and resolve this for the Threatened Species Initiative. And what we decided to do initially was to simply say, let's try and solve an initial challenge of how do we get data from that's sourced on the BPA, the Bioplatforms Australia data portal, raw sequence data, easily send it to Galaxy Australia and analyze that data. So how we formed up the challenge and how we formed up an initial picture of what we wanted to do was to simply say that as a researcher up here, you want to engage in this or connect to this platform called the community portal, a single entry point. And then from that point, you have access to everything you need in terms of documentation, in terms of links, and the ability to access other services like the Bioplatforms data portal and Galaxy. And of course, the ability to be able to send or ship data from the BPA data portal to Galaxy. The The key thing to note here is that the community portal was a specific request from the Threat and Species Initiative because they really wanted a one-stop shop for all of this infrastructure. Part of the challenge is that everything is dispersed, documentation, access points, links are also dispersed. There's no coherence amongst those various services and platforms. So this idea really also responded to not just making research easier, but subsequently, where does one start, and making that starting point super easy and super straightforward. So why would we have a TSI service portal or a community hub? We're still working on the name. Well, it's a central online hub from which users can jump to different resources and services easily. So as I said, it's like TSI data on the BioPlatform's data portal. It's like accessing assembly and annotation pipelines on Galaxy, and also being able to access documentation and how-to guides and help as well. The technology we developed also provides single sign-on across these multiple services, and some other nuances and some other important things like the management of membership and access rights to data and services, because TSI is one of many different communities. There's Genomics of Australian Plants, there's Oz Mammals, there's Zero Childhood Cancer, there's many others, and they will each have their own rights and privileges, different types of data and different styles and types of resources. And ultimately, having the TSI service portal is, as I said, just about making the process of research much easier. So we worked with AEF to build a functional proof of concept, which provided a single entry point. It provided researchers to access the BPA data, the ability for researchers to access the BPA data portal and to be able to search for their data of interest. To be able to very easily transfer data from the BPA data portal to Galaxy, to then be able to log into Galaxy and process data. And then some other kind of extra benefits around outcomes around being able to self-register via a login of your choice, whether it's your institution or Google, etc. And then to be able to also apply and request TSI community membership. And to be able to use that that identity or that account to be able to access these multiple services. And as a community manager, so as someone like Carolyn Hoggs, to be able to also initiate membership, to be able to say, I met Stephen Manos at a conference and I would like to invite him to be part of this online portal, and being able to go through that process to invite them along and have them ultimately approved to be able to join that community online. So I'm going to give you a live demonstration now, and this is really just focused around demo number 1. And the user story behind demonstration number 1 is, as a user, I want to be able to easily find data and easily send it to Galaxy for analysis, all guided by clear documentation. We have some other demos, which I won't show today because we're limited on time, but it's around the user onboarding and access management. And the user stories there are that as a user, I want to be able to easily register for and get access to multiple services. And the second story is, as a community manager, I need to be able to invite people to the platform and manage access in a straightforward and scalable way. I'm not going to show demo number 2, but they are also things that we have worked on over the last year and a half. You can assume that for the demo right now, I've already done 2A. I've already registered for the service. I've already had my account and my registration approved. And now I'm logging in for the first time and getting access to these multiple platforms. So I'm going to go ahead and start with a window here on our Directus website. Directus is a CMS. And this is the starting point. This is the single starting point that I described that was a requirement of our from the researchers we were working with in the Threat and Species Initiative. So here I have the opening page. I go sign in with CI Logon. I can select a number from a number of different identity providers. Here I'm just going to go with Google. I'm going to log in. And I've already signed in as my identity that I'm using for this demo, called test member 584. So here's the view of the community portal or the service hub, and I have access to various data sets. This is the region Honeyeater here. So this is information that's being pulled down from the BPA data portal. I have access to various tools on Galaxy. There are various user guides that I have access to that guide me through the process of running assemblers on Galaxy Australia. And I also have ongoing access to various services that are connected via this single sign-on methodology. So I'm just going to go ahead and access these services. I'm going to click on the BPA data portal link here, so you can see that this is going to a completely separate service now. I click login, single sign-on. Again, I'm going to log on via Google, and it should just drop me into this same identity. So what's happened here is through the process of registering with the Community Hub, I've also had my identity created within the BPA data portal, and I've also similarly had my identity created within Galaxy Australia. So I can go back here to the online portal. I click on Galaxy Australia, log in, and I get logged in as that user again. So what I want to do now is I want to be able to go to the next step is to go to the BioPlatforms data portal, or even easier, just go to the hub, the service hub here, and which will automatically link me through to the region Honeyeater data set. Generally from here, in the past, the process of getting data from the BPA data portal to something like Galaxy meant finding this data, downloading it. This here is a small data set. It's for test purposes, but they are much, much larger typically. And downloading these over wireless onto your laptop, getting it back into the BPA data, into Galaxy or other platforms for analysis, can be a very arduous and error-prone process. So here what we've done is decided to just make this super easy. You click access, you go send to Galaxy. It says resource sent to Galaxy. You move over to Galaxy, and you can see that Galaxy is now in my history receiving that BAM file. So it is receiving that genome sequence, and it's done. And it is literally a one-click process. And in the background, this is using the GF4GH DRS standard to be able to link those two resources together and send this data across. As I said, this used to be a very complicated process. It's now a much, much simpler process. I can now refer back to the Science Hub here around finding guides and anything else I need to proceed with my work. From here, I can simply say, well, I need to now run this workflow. I click on workflow here. This is the one I want to run against my data. This workflow is smart enough to know that my most recent uploaded sequence file is the one that it should use. I click run, and then it will, and then I click run workflow, and then it will begin the process of invocating the various steps of this workflow. And as you can see here, it is quite a complicated multi-step workflow across various individual tools to make this assemblage happen. So just a quick recap. I've given you a quick demonstration and overview of our work with the threatened species. We have, with the AEF, developed a proof of concept of this idea of integrating these services. The aim was to really hide the complexity from researchers. I should say that there's a lot of code and configuration and customization behind the scenes. We've endeavored to use standards like DRS and OIDC to make all this work extensible. And I will, I guess, finish by saying that a lot of the work here requires not just effort on the access and identity side, but also on the application side. And it's true to say that most of the effort has gone into our work with CCAN, which is the technology behind the data portal, but also Galaxy Australia, because those code bases needed extensive modification to make these access and identity and also data insertion aspects work. And we've had some great successes lately because all of those changes have been, generally speaking, accepted upstream, and particularly for Galaxy. And our next steps around this project are really to continue this work and aim to bring this idea of a service portal for not just the Threat and Species Initiative, but also for other communities as well, into production in 2024 and beyond. So thanks everyone for listening.
We'd like to thank Stephen for putting together this presentation, and I'd also flag that we do have Nigel Ward here today who worked on this project, and he will be able to answer any questions that you may have.